Here we are at the HUPO World Congress in Toronto, and the new version 25 of the Human Protein Atlas has just been released. Tell me about this new version.
We are very excited about this new release with a lot of new data across the different parts of the human body. Researchers can explore the different tissues and organs and locations inside the cell and so on. Altogether, we now have more than 10 million images, annotated images, and actually 6 billion data points using 27,000 antibodies. So I'm very grateful for the more than 1,000 researchers that have been formally involved to create this open access resource.
So what is new in this new version?
Well, first we have again used the Nobel Prize winning algorithm from DeepMind in London. And in this launch, we show the predicted 3D structures of interacting proteins using the AlphaFold 3. Altogether, actually, 23,000 protein interactions have been modeled, and it is now possible to visualize these structures in different ways, including where our antibodies are binding to the structures. Secondly, we have made a huge effort to extend the data on single cell and spatial transcriptomics, where we want to show the RNA levels both in tissues and cells. This is really an excellent complement to our single cell antibody efforts. And we now have included a lot of new data from new parts of the human body, such as adrenal and pituitary gland and urinary bladder. We actually have data for more than 1,000 tissue-specific cell types, and we believe that this is a fantastic resource for all researchers interested in the human building blocks of human biology and disease. Last but not least, we also launched a new updated and extended version of the human disease blood resource. It covers patients with more than 60 different diseases to allow the research community to explore what proteins are elevated or decreased when you have a specific disease. This new resource is built on 3 complementary assay platforms that could be described as sort of next generation blood profiling. We're very excited about this. The assay are actually proximity extension assay developed by the company Uhlen in Sweden. It is SomaScan developed by the US-based company SomaLogic, and also the target proteomics using mass spectrometry developed by Proteome Edge, also a company from Sweden. In this launch that we published today, it is possible then to compare the blood profiles from more than 60 diseases around thousands of protein targets. We believe this open access resource will be important to avoid some of the reproducibility issues that has plagued biomarker discovery and precision medicine for years.
What issue are you talking about?
Oh, it is, of course, a huge need to find biomarkers that can tell the physician if a person has a particular disease or how to treat that person when he has that disease. This medical need, which is enormous, has resulted in a lot of scientific literature, an explosion actually. And in fact, several articles are published every hour with biomarkers. Despite this, very few biomarkers have made it into the clinic. There are several reasons for this failure, and we have tried to address some of this in this launch. First, we have used next generation blood profiling, which are both comprehensive and sensitive. Secondly, we use a pan-disease approach to address the overlap of profiles between different diseases. And thirdly, the fact that proteins are analyzed with at least 2 independent assay methods means that we can validate the data already sort of when we are doing the discovery. So the fact that all of this data is now available hopefully makes it easier for people in the future to use this in their own biomarker projects.
Congratulations on your new version of the Human Protein Atlas. Do you have any final remarks?
Well, thank you. I want to thank the Knut and Alice Wallenberg Foundation supporting the HPA project now for more than 20 years. And I'm really grateful that they have supported the decision to make this data open access to allow researchers around the world to explore this wonderful world of the most important building blocks of human beings, the proteins.